talk here. My name is Ruben Garrido-Oter, and I work at the Max Planck Institute in Cologne. And actually, I don't know if I can. Oh, thanks. I wanted to give first a general sort of introduction to the model systems I work with, but I think it might be a little bit redundant now with respect to the talk we just had. But I can start maybe from where I started, which was actually not cyanobacteria, unicellular photosynthetic organisms, but actually embryophytes and land plants. So as you probably know, when plants grow, they alter the physicochemical properties of the surrounding soil. In essence, they secrete a cocktail of compounds that include organic carbon derived from photosynthesis. And this cocktail of compounds is called root exudates. And when they do so, they essentially create a niche around the root, which is called the rhizosphere, this area under the influence of these exudates. They create a niche for colonization by environmental heterotrophic bacteria and other microbes such as fungi. So these microbes assemble into well-structured communities that we call the root of the rhizosphere microbiota. And in exchange for carbon, they provide the host with beneficial functions such as nutrient mobilization, protection against pathogens, and so on. And now there is more and more evidence suggesting that these microbial communities are absolutely essential for the fitness of the plant host. And what is interesting to notice, but you already know, is that these processes not exclusive to multicellular photosynthetic organisms, but can also be observed in unicellular ones like microscopic algae, which also can secrete photosynthates to the area surrounding the cell, which is called the phycosphere. And they also assemble complex microbial communities, which are called the phycosphere microbiota. So these interactions are very well studied in aquatic environments where we know they play important roles in global carbon and nutrient cycles. But although microscopic algae are also very abundant and found in terrestrial ecosystems, particularly the soil surface, how they interact with soil-derived microbial communities and how this process is analogous to how multicellular organisms such as land plants do so is not well understood. So I'm mostly interested in studying how host microbiota associations have diversified in the green tree of life. And I started, as I said, looking at the root microbiota of different plant species. One of the ways in which I did this is now a couple of years ago, I collected amplicon community composition data from roots of plants from a very wide variety of species. So this is a meta-analysis collecting from different studies. And you can see that they actually include a very wide diversity of plant hosts. And I use some experiments that we performed in my lab using the soil-borne model chlorophyte algae Chlamydomonas reinhardtii. And what I did was to compare all these microbiota structures, and one of the main results from this meta-analysis was the identification of a small set of bacterial taxa, which are found in associations with all these very diverse hosts, independently of the soil type and the biogeography or the lineage of the host. And that importantly, this core taxa could be extended to the phycosphere of these soil-borne algae, indicating that there are perhaps some conserved patterns. And early this year, we published from my lab, we used the model Arabidopsis thaliana and again Chlamydomonas to do some comparative microbiota experiments. And we actually identified a significant overlap between the root and phycosphere microbial communities. So this, of course, suggests that there might be some core mechanisms, core ecological or molecular or genetic mechanisms that explain some of these common patterns of associations, at least in terrestrial ecosystems. And I'm interested in studying these processes, so we further develop this model based on green algae, and we do this using reductionist experimental systems. So these are based on culture collections of microbes and what we call synthetic communities. So the idea to explain briefly the outline of our sort of experimental design, our experimental system, is to start from an environment. In this case, we look at phycosphere communities from terrestrial environments. Then we start by isolating as many microbes from these environments as we can, and we have different approaches to do this, but the idea is to try to capture a culture collection of isolates that represents as much as possible the complexity of the natural environment. And then there is a next step which, informed by what we observe in nature and using our culture collections, we have, from hundreds to thousands of these isolates, we can reconstitute them using autobiotic systems. So it's a highly reductionist experimental system. And the idea is that we design these synthetic communities or syncoms based on observations from natural communities and with the goal to test hypotheses. And the objective is that what we learn from these reductionist systems can inform and generate new hypotheses that can be tested and so on. So we did this for the model Chlamydomonas, and we started with experiments that are very simple, look like this. We have arsenic, algae, and soil-derived, so bacterial washes, soil-derived communities. We can grow them alone, without carbon, with organic carbon, or we can grow them in co-inoculations with the green algae. So to cut a long story short, this is published data. We published, as I said, earlier this year. What we find is that when we co-inoculate these microalgae with soil-derived bacterial communities in the absence of external carbon, there is microbial growth of a specific phycosphere community, which are the samples you can see here in green, that is independent also of the stat inoculum, the soil type, and the media that we use. And what we can see is that if we calculate ASVs, so amplicon variants that are enriched in these associations versus those that we find in soil, these are the green ones, are the ones that we find enriched in associations with Chlamydomonas. And you can see how in our initial soil washes, they are barely detectable, but very quickly in co-inoculations with the green algae, they grow and they come to dominate the community. So there's a process of recruitment of very low abundance microbial members from the soil community that in interactions with these phototrophic hosts, they really grow and assemble into a distinct community. So taking samples from the system, also from soil-based systems, what we did was used a combination of approaches, mostly relying on dilution series, and we isolate the culture collections from this environment. And this you can see a rank abundance plot from the most abundant ASVs that we find in the natural communities. And in black, you can see those for which we at least recovered and isolate from our collection. And this you can see a phylogeny of these most abundant members from the root and rhizos phycosphere, but the phycosphere you can see here in green. So essentially all these green squares represent an abundant member in the phycosphere community. And those squares with a black outline, those are bacterial species for which we have at least one representative isolate. So we have sequenced all of these isolates, and now we can take our culture collection. We have 196 validated strains that we have sequenced, and we can design synthetic communities, and together with the succinic host, we can grow them in autobiotic system, in this case, for example, in bioreactors. And the idea is to do this for in a closed autobiotic cycle so that we can derive very rich time series from these very simple synthetic systems. So to do this sort of experiments, I will show you an experiment that we are analyzing at the moment. So we use a type of bioreactor that perhaps you're familiar with. It is called a turbidostat. The idea of this is an autobiotic system where we have fresh media without carbon, and the system maintains a constant cell density. So that's why it's called a turbidostat. It monitors optical density and it pushes fresh media to keep a constant density. So it simulates an infinite environment for growth of these phototrophic consortia. And this all carbon comes for photosynthesis, and this is a minimal media. So this is how it looked like. We repurpose such a system, and we managed to basically keep it close and run it autobiotically. So this is work from Pepe, a postdoc in my lab, who managed to run 8 independent replicates with a 22 bacterial synthetic community over 125 days. So we actually thought that the system was going to collapse after a few days. Perhaps a fast-growing bacteria member will dominate the community, but in essence, we can keep observing we can observe a complex community over the duration of the experiment and no significant loss of diversity. So when we analyze this, it's all amplicon 16S sequencing. So we take samples and we do shallow amplicon sequencing every day, essentially. And we can do this because it's a simple member. We only have 23 members, so we don't really need to sequence very deep to obtain community profiles. So what we see are these types of interactions, and this is an example where I highlight Chlamydomonas abundances in green versus total bacterial abundance in gray, and you can see that there is some patterns of complex interactions that oscillate over time, but not only between, in this case, the primary producer and the heterotrophs, but also between individual members of the microbial community. For example, these two bacterial strains that are highlighted in color that show these patterns of stable co-exclusion throughout the experiment. So when we analyze this time series data, and again, this is only community composition, we can derive a network of metabolic interaction. And we see some positive interactions here in red, but interestingly, some of these negative interactions between, for example, these two strains that are partially mediated, we think, by production of antibiotic and antibiotic resistance. But some others that are a bit more difficult to explain, for example, these flower bacteria that actually has negative interactions with pretty much many members of the community, but nonetheless is not in binary association, doesn't seem to be able to inhibit the growth of the other. So this probably reflects cycling in the available pool of organic carbon. So of course, one of the questions is, if you, after so many generations of bacterial and algal growth, do we see some footprint of artificial selection in these synthetic communities? So for this, what Pepe did was to take samples near the end of the experiment, I think this is day 122, isolated the bacterial fraction. This can be done either by centrifugation or filtering, and then using a cell counter estimated a way to plate dilution series of these samples into plates containing rich growth media, which then are incubated. Then we can sequence, do 16S sequence of these re-isolated strains. And in this case, what we did was to sequence with PacBio these evolved or these re-isolated strains. So by analyzing these genomes, we did this is basically, I think we re-isolated and resequenced 22 of these bacterial members. And we see a variety of variants appearing in these genomes. But interestingly, and much to my surprise, and this was one of the motivations to do PacBio, because I thought that in a community context, the strongest process for selection and for evolution would be horizontal gene transfer. Or actually, we didn't find any signature of horizontal gene transfer in any of the resequenced members. So what we did find was variants appearing in categories which actually make quite a lot of sense in such a system. So this is also typical from artificial selection experiments. We find mutations appearing in sigma factors and global regulators, also in motility genes, some things that might be related to the metabolic interactions with the algae and with other bacterial members, such as central carbon metabolism genes, but also some, for example, some antibiotic production and resistant genes that can explain some of these negative interactions. So of course, what we want to do now with these re-isolates is to perform experiments by comparing the phenotypes of ancestral versus evolved strains. And of course, now we have started a series of new experiments with different conditions, and we will also do a shotgun of different time points to really see all the standing variation in the system. So with this, I would like to finish and thank my group, in particular Pepe, who has been doing most of the work that I showed you today. Yes, and thank you for your attention.
Thanks a lot, Ruben, for a nice talk and keeping on time. So I'm going to take questions. If you can briefly repeat the question for the people on Zoom, that will be great. All right, any questions? Any?
You compared monoculture evolution with your community evolution. You see when mutations are shared and not shared?
Yeah, no, we haven't done that. It was basically, we just wanted to assess after so long how how much variation we will observe. But that's a good idea. We can. What I was thinking about is to have two parallel tracks, so to speak. One bacterial community that is growing with chemical complementation of organic carbon, and another one that is growing with photosynthesis, with co-inoculations with a primary producer, and then just compare these two communities in terms of their selection and evolution.
All right.
You find that your system is evolved, I think, and then have you compared the interaction network with the change after the evolution? For example, I think in your results here are some antibiotics and also secreting system changes. Do you think that it means the increase of competitive impact?
We haven't done this analysis, this comparison analysis yet. So an advantage, of course, of having re-isolation and evolved strains is that we can now reconstitute the same system side by side with using ancestral strains and compare their behavior. Yeah, but we haven't done it yet.
I'm wondering, you infer from the correlation whether interaction is positive or negative. You try to do it in a sliding window to see if you can find signatures of changes in that correlation as a function of time.
Yeah, I looked a bit into this, just trying to compare networks inferred from different epochs, so to speak, of the system, and they are, in my opinion, they look rather robust. These interactions don't change a lot over time, but of course, there is it depends on the size of the window and so on. But I think they are more robust than I would have expected. Yeah, perhaps we need to run this for longer to see significant changes.
What I mean is that actually seeing that it breaks from robustness means there's an application that changes the interaction. That's a signature of a change in interaction.
So by robust, I mean if the comparison of networks inferred from different epochs, they are very similar. So that to me was surprising. But we haven't really done a very systematic analysis of this.
And maybe to follow up on the networks, was a bit technical interactions for this covariance.
Yeah.
So did you have any evidence in your genome that you sequenced that shows that there are actually interactions?
We are looking into actually, rather than from the genomes, from binary association experiments. For example, some of these Pseudomonas members, they really antagonize the growth of many others. So we think that maybe obtaining phenotypic data with small communities and high throughput assays will allow us to go from this, as you're absolutely right, these are just co-occurrence or co-exclusion patterns, to go from there to hypothesis about what mediates these interactions. Yeah.
Okay. Yeah, Catherine.
I wonder if you have total abundance data for your you've got relative abundance data. Do you have something on total bacterial load?
Sorry.
Do you have like total 16S qPCR for total bacterial load? I ask because I think about your surprising flavobacteria interaction, where you don't see it, where if you're just looking at relative abundances, this might come from a confounding factor that as some things go up.
Yeah.
If it's a weedy grower, it goes down, and that's driving what you see.
Well, keep in mind this is a turbidostat, so cell densities are constant. There's in principle there are the same number of microbial cells per milliliter all throughout the experiment. So these are relative abundances, but in some way one could interpret them as absolute abundances.
In that scenario, do you have any way of tracking like flow? Because you will modulate flow in and out, presumably. So if things are growing more, you presumably sweep more through the system. So you could still have a notion of, if you were not keeping things the same, some things would be growing more. So poor old flavobacterium or whatever goes down. And do you have that data that you could incorporate it into your interaction network and see if it still holds?
I think a very interesting parameter to play with is precisely this OD threshold. So to have a system running at a lower cell density or a higher cell density, I think some modeling work shows that this will have an impact in some features of the system, for particularly the chaotic behavior, the predictability of the system. So now we are doing some experiments with more replicates where we have different densities. We have the same starting microbial community, but we grow them at different sort of growth rates, because essentially that's what it ends up being, but at different densities, and hopefully we will analyze the differences in behavior as well.
All right, one last one, Chris.
So I guess it's a related question, but why do you choose stabilistat rather than clinostat?
Because it's in some ways they are easier to run. We can run 20, 30 of them at the same time. I think that would be very difficult with a proper chemostat. But I would ideally, yes, we would like to do that. Yeah.
Right, I'm going to take one more question while Amelie gets sets up,
Just
if that's okay.
Yeah, if the ratios from your community are the same in the beginning, and then this cell density allowed some kind of threshold for the variability of the community.
If the threshold of optical density,
really for the starting, let's say it's just it's been said that in microbial community sometimes the proportions of the components of the community could have an impact. So to know what was the starting proportion between.
We haven't tested that systematically with the system, but you know, after I guess we started doing synthetic community experiments also with plants, you know, over the past 5 to 7, 8 years, I think every experiment that we have performed with different starting ratios, they typically converge into very similar communities. So at least for associations with plants, and I suspect also for microalgae, the starting ratios are not so relevant. And you can see how at the beginning we have some sort of behavior which is, you know, we find in more replicates there is a very fast growth of a fast-growing Pseudomonas that then, you know, kind of goes away. So I think these initial ratios are, so the communities typically converge to something else. Yeah.
All right, let's thank Ruben and all that. All right, we have to do a little bit of sort of dancing on our feet here. So I got a message from Otto that